How to download a nucleotide sequence from Genbank. The National Center for Biotechnology Information (NCBI) is one of the world's premier websites for biomedical and bioinformatic research. It is a part of United States National Library of Medicine (NLM). NCBI maintains the Genbank Nucleotide Sequence Database, which is an annotated collection of all publicly available DNA sequences. Genbank is part of the International Nucleotide Sequence Database Collaboration, which comprises the DNA Data Bank of Japan (DDBJ), the European Molecular Biology Laboratory (EMBL), and Genbank at NCBI. These three organizations exchange data on a daily basis. To download a nucleotide sequence from Genbank, we begin by a Google search of NCBI. The search results appear. Select the first search result, which says National Center for Biotechnology Information. This leads us to the home page of NCBI. In the home page of NCBI, we see a drop-down box and a search bar. Click on the drop-down button and from the list select nucleotide sequence. If you are searching for a protein sequence, you can set the option to protein. Now we are selecting nucleotide sequence and type the name of the sequence that you require. Here I am typing SARS-CoV. Click on the search button. The result page appears, and here we can see that the result page contains 20 results per page. Out of 24,000 results, the page shows 20 results. Scroll down through all the results and you will find that the results appear in a pattern with title, the length of the nucleotide sequence, type of the nucleotide molecule, linear RNA molecule here. It also gives the accession number and the gene info identifier. The results also shows that the gene sequence is available in Genbank, FASTA and graphic formats. Scroll through the results and select the most appropriate result. The Genbank file appears. So this is the BAT SARS-CoV complete genome in Genbank format. Before downloading the sequence, let us look into some of the important fields in Genbank flat file. Genbank flat file is a term used to describe the downloaded sequence. The locus field contains a number of different data elements, including locus name, sequence length, here it is 29,000 base pair, the molecule type, here it is a linear RNA molecule, the 3-letter code VRL shows a Genbank division. VRL here stands for viral genome and the date is a modification date. The next field is definition. It gives a brief description of the sequence, which include information such as the source organism, gene name or protein name, or even description of the sequence function. Here the source organism is BAT SARS-CoV. It is a complete genome. The next field is accession. It's a unique identifier for a sequence record. An accession number applies to the complete record and is usually a combination of letters and numbers. Next comes the version, a nucleotide sequence identification number that represents a single specific sequence in Genbank database. Even if there is a single nucleotide change in the submitted data, the version number will be increased. Next one, keywords. Usually words or phrases describing the sequence are given here. The keywords are not mentioned in the sequence, so here it is not left blank, but a full stop or a period is placed instead. The next field is source. It is a free format information including an abbreviated form of the organism name, sometimes followed by the molecule type, etc. Organism. The formal scientific name for the source organism and its lineage based on the phylogenetic classification scheme used in NCBI taxonomy database is given here, and it is shown as a hyperlink which leads to the NCBI taxonomy database. Reference. The publications by the authors of the sequence or those who deposited the sequence in NCBI, which deals with the data reported in the record. Next field is authors. It shows the list of authors in the order in which they appear in the cited article. Title gives the title of the published work or tentative title of the unpublished work. Journal. This field contains the Medline abbreviation of the journal name. Scrolling down to features. Features include information about genes and gene products as well as regions of biological significance reported in the sequence. These can include regions of sequence that code for protein and RNA molecules. It includes source, which summarizes the length of the sequence, scientific name of source organism, taxon ID number. It includes taxon, which is a stable unique identification number. It includes CDS or the coding sequence. Scroll down and you will find origin. The origin may be left blank and it usually indicates the sequence start. So that's all about the nucleotide sequence in Genbank format. Now we can move up and click on the FASTA hyperlink so that we get the FASTA format of this nucleotide sequence. A sequence in FASTA format begins with a single line description followed by lines of sequence data. The description line is distinguished from the sequence data by a greater than symbol in the first column. It is recommended that all lines of the text be shorter than 80 characters in length. Now, to download or copy the nucleotide sequences, we can either choose Notepad or Microsoft Word. Here I am selecting Notepad. Open the Notepad window. Then copy the FASTA format sequence. The complete sequence, copy and paste it to the Notepad. Now save it and your sequence is available offline. You can also repeat the same to a Word file. The third format of nucleotide sequences available in Genbank is a graphic format. The graphic format looks like the following.